Alright, welcome back. Hi AP Bio. This will be my last video for today, close to the end of the PowerPoint of sample FRQs, but I do have some more for you that I'm trying to hunt down. So if this is really helpful, let me know in the comments, send me messages. If you want more, I've got a couple more coming down the chute. So this, I believe, is going to be part 6, I think. Ooh. Okay, the last one, because it's almost 3 a.m. and I don't know, I'm just still going and I thought I'd finish it. So I am now in practice exam number 2 for 2020, and I'm going to be going through number 1. Number 4 from that test I'm not going to do because I just looked at it and it covers mainly ecology and evolution. This is it right here if you would like to pause the video and look at it. It's also in the AP Classroom, but this has a lot of content that is not covered on this year's exam, so I'm not going to bother going through that one. So number 1 is all about cellular energetics, cellular respiration, fermentation, and our electron carriers. So actually, I'm going to, what am I going to do? New slide. I'm going to do something live here. It's probably going to make me run over my time, but I think it's a good idea. So I'm going to make this really big. And I'm going to make this guy really big. So we talked about cellular respiration and its components a while ago, and this question is a great review of that. So just what they're doing here, remember the NADH and NAD plus are the electron carriers. They're those little trolleys and shuttles that are carrying the really excited electrons from these oxidation reactions, right? Oxidation, Leo the lion goes grr, lose electrons, oxidized for Leo. So glucoses, fatty acids, proteins, and other organic compounds are losing their electrons by oxidation, and those excited electrons are hopping on the shuttle here in glycolysis, also in the Krebs cycle inside the mitochondria, but their end destination is like the Disneyland of the electron transport chain. That full shuttle with those excited electrons is going to dump the electrons off into the electron transport chain, and their final destination is here to bond with oxygen gas to make the water that you breathe out from cellular respiration. So what is going on in this question is that there is an enzyme, where is it? NADH dehydrogenase enzyme. So NADH has its hydrogen. To dehydrogenate something is to take a hydrogen off, and ACE is an enzyme, okay? So the enzyme NADH dehydrogenase is basically helping here in the electron transport chain to do this arrow right here, okay? To get the electron off of the NADH, that electron goes through the ETC to help charge up, the electron excites these proteins to move the hydrogen proton across. So when you get an NAD plus, you also get an H plus because the electrons are traveling, and they relax after they've done all that pumping work and help, and they then bind to the inorganic low energy water with oxygen. So what's going on here in this question, I had a whole bunch of notes. This one was pretty detailed, so I don't know, you probably can or can't see this. Nope. Boop. Who cares? I was basically showing that the mutation people, they don't have that arrow, or it's not working really well, right? It's not releasing, and so where do these shuttles go if Disneyland is closed? They drive around in the parking lot, remember? Like they go to lactic acid fermentation so they can recycle and get back NADHs to keep oxidizing things, but you don't really get the ATP out of that, but you do get your empty shuttle back. So people with this mutation in this mitochondrial gene, MT for mitochondrial, NADH5, right? It's clearly titled for a reason. They have a buildup of lactic acid. So rather than offloading here in the presence of oxygen and being able to do the electron transport chain, they're doing fermentation whose product is lactic acid. So that's not helpful. The researcher is hypothesizing that the mutated enzyme has decreased activity but is not completely non-functional, right? If you can't make any ATP ever, like, you'd probably be dead. So it's just worse at working, okay? And that increasing the pool of NADH in cells, so providing more of this, might increase the activity of that enzyme. That's this scientist's hypothesis here. So remember, in a question 1, you've got an experimental design. You're going to answer stuff about content. You're going to answer stuff about the design of the experiment, predicting things and changing in the future, and then kind of some justifications of those claims. Here's the graph. So to test this idea, researcher treated a group with a vitamin that is similar to the substrate that it acts on, right? So we're starting here. We're basically providing something else that kind of looks like this. So can we still go across if we have a supplement of this will the faulty enzyme work on that thing? A slightly different kind of shuttle provided as a vitamin supplement. And they measure the concentration of NAD plus as a result. Okay, so they've got that here, and they're also measuring the lactic acid. So if they, I totally brain fart right there. Oh, that's right. They're measuring the concentration of like the product at the end, right? Because if you can turn this into this, you're able to do one of these. So they're measuring not only the NADH, but also the product of fermentation to see which processes it's changing in. Okay. The results of a representative individual are shown here. There we go. So here are your questions. This is a 25-minute question, so at this point in the video, if you wanted to pause and stop, read these questions, and work on that, go for it. And that's what this slide is intended for. This one will have the link to this video, and then let's go through it. Half my time, I'm going to race here. Here we go. So describe the pattern of inheritance that is most likely associated with mutation in the MT5 gene. Explain why individuals are not typically heterozygous with respect to mitochondrial genes. That's the clue here. So pattern of inheritance, where are you getting your mitochondrial genes from? Remember the song we listened to, like you're a little bit more your mom than you are your dad? It's because you're getting it from your maternal parent. So the mutation will only come from mothers, not from fathers. So you could say anything like that. Describe the pattern of inheritance that is associated with the mutation. We're getting it through maternal parent. Mothers are passing on to all of their children, anything like that. And explain why you can't be heterozygote for a mitochondrial disease or mitochondrial gene. It's because you only got one set of mitochondrial genes and they were from your mom, so you only have one chromosome copy of these. She's not carrying 2 copies of different kinds of mitochondrial chromosomes. So I would also say only one gamete carries the mitochondrial genes, the gamete from the mother, so one cannot be heterozygous because only one mitochondrial gene set is inherited. You can't get like being mutant and non-mutant. If you're mutant on that gene, that's it. So that one is A, that's 2 points. B, identify the dependent variable. These are very easy. It's reading off the graph. So you're looking, it could have been both, except one of the following. They didn't ask for both. Identify a dependent variable. So if you're looking at the graph, what is it that they're measuring as time goes on with this vitamin supplement? What are the concentrations of either of these two things? I don't see that you have to include the units on there. Lactic acid, NAD+, what's the concentration in the blood? That's there though. So look, my kitty's snoring. Yep. It's a snoring Jackson. That's adorable. Okay. Totally distracted now. So if we go back up oh gosh, not too far. Concentration of NADH, lactic acid in the blood. Oh, excuse me. So maybe that would be pertinent to mention. Make sure you're catching all the vocabulary of what is in the dependent variable. Identify one control that the researcher could use to improve the validity of the experiment. Now, you guys are all probably old enough to recall the biology EOC and the validity and reliability and how do you improve either. It's not as stringent here in AP. I think it's terrible. Like, if you look up synonyms of validity, reliability is the synonym. It's terrible to use these two terms, but reliable, I always think repeat the experiment, do it again, getting the same results, then it's reliable. Improving validity is making it better, designing and adding another element of your experiment to confirm your results are happening because of what you did. So measuring NADH and lactic acids in a group that is treated with a placebo, I was thinking just measure individuals without the vitamin. That could work too. I don't think you have to use placebo, that fancy word. Placebo is like a water pill. It's a pill that has nothing in it, but they think that they've gotten the medication. And lastly here, justify the researcher analyzing blood samples at any intermediate time points instead of only at the beginning, at many, intermediate, instead of just doing it at the beginning and at the end of the 20 weeks. I think this one's a real gimme. You get to see a more accurate trend, you get to see more data. I thought you could see if the effect wears off or is sustained a certain amount of time that the vitamin works and then stops working. And so I think this whole question, part B, was pretty simple for 3 points, like out of a 10. So a lot of these questions, like, you're going to be able to knock them out real quick. In C, describe the relationship between the concentration of NAD plus in the blood and the concentration during the first 5 weeks of treatment with the vitamin. So this one's pretty simple. Describe the relationship. You're going to look at that graph. They're not asking you for numbers. As one increases, this one decreases. Make sure you get them correctly written out, right? And that's exactly what they have. The concentration of NADH increases and the concentration of lactic acid decreases. Make sure that you're looking at the right time frame, first 5 weeks, okay? Based on figure 2, calculate the average rate of change from 5 weeks to 17. So now you got to get, like I said, some kind of ruler to put on your computer screen or a straight edge, like a piece of paper or something, so that you get this number right. I actually thought this number was 50. It's really late and I'm tired, and it's actually 60. So here you're calculating the average change. It's a slope calculation, rise over run. So we're all way out here at 60. What's the difference between 60 and 300? And 17 minus 5. Oh, that's not in the way too much. And the answer is 20, and it looks like they're accepting both 20 and negative 20. So, oops, I should not move that. It's not a math class, so I'm guessing the negative slope is not important. So, and the last one, we got to race for time. All right, probably not going to make it. You know what? I'll stop here and then accurately do D before going too fast. Okay, so that's A, B, and C of 2020 practice test 2 FRQ number 1.